The main mission of our division is to carry out outstanding basic research, to discover new genes and their functions, and then ideally to figure out how those genes can be translated into clinical and medical practice to help patients. One of the remarkable features of the activities in our division is their interdisciplinary nature.
The thing that makes the Division of Genetics at Brigham Women's Hospital unique is the diverse expertise. People work on genetics, but they use different types of approaches to ask different types of genetic questions.
This is really one of the few genetics divisions that is really embedded in a department of medicine. The close connection to the clinical aspects of our work is really highlighted essentially in any of our projects.
The Brigham Genetics Division has great strength in cancer genetics. We have a new Center for Targeted Cancer Therapeutics led by Dr. Karen Schakowsky.
My laboratory is interested in understanding how the RAS pathway drives tumor development. Recently, through genomic studies, we identified a new tumor suppressor that cooperates with the NF1 tumor suppressor. And from this genetic analysis, we were able to identify therapies that target each of these two mutations and found a combination therapy that really helped cause these tumors melt away in our animal models.
Another area we work on is aneuploidy in cancer, and that's an abnormal number of chromosomes. We were able to build a mathematical model that actually predicts which chromosome aneuploidy events are actually driving cancer.
In our laboratory, we are using zebrafish to study formation and regeneration in cancer formation. One of our projects, we have analyzed a family where multiple members were afflicted by pancreatic cancer, and we're able to find a gene that we think is causative to put this family at high risk for pancreatic cancer, and have now made a model in the zebrafish that is also cancer prone.
My group is collaborating with a pathologist from Brigham. We're working on a particular kind of squamous cell cancer where there's a fusion that's caused by genetic translocation, and the fusion brings together two proteins that would normally not be together.
Brigham Genomic Medicine is an exciting new integrated clinical service line where we take patients with undiagnosed diseases who have an underlying genetic basis, sequence the genomes of those patients, and using computational approaches figure out what the causal DNA sequence variants are.
Division is strong on computational biology and bioinformatics. My program revolves around genetic variation. So we're interested in all questions about genetic variation, about the origin of mutations, try to find statistical patterns and trying to generate hypotheses about the origin of mutation.
The genomic medicine program is the place in our hospital and division where basic research meets the clinic. We have about 25 world experts in clinical, computational, and experimental biology that meet together on a weekly basis to solve undiagnosed Mendelian cases. We start by case presentation and assessment. The clinical geneticists show us the clinical presentation, the family history, and we together hypothesize who are the most informative individuals in this family that would serve for genomic sequencing.
Our work in a powerful new imaging technology called multi-isotope imaging mass spectrometry, or MIMS for short.
MIMS allows us to peer inside of individual cells and really inside organelles residing in individual cells. We can also perform high-resolution, very precise functional measurements, and we can do that at a nanoscale that really nothing else can achieve. One example was in the hair cells of the inner ear, where MIMS was used to look at protein turnover in these small stereocilia which project into the inner ear. Another example, we looked at heart regeneration in rodent models, and we didn't just quantify how much heart regeneration was happening after the heart was injured. We were able to dissect where new heart muscle cells were coming from.
Genetics just keeps getting better and better.
What I think is so exciting about this division is that the leadership has had the vision to not have a narrow view of what human genetics is.
We're really in a rebirth of human genetics and the ability to understand the human genome.
When the genome was first sequenced, there were expectations that the information would be applicable to the health and care of patients. Now, a decade out, we're now in the position to finally do that, and that's proven to be very exciting.